Welcome to the second video on amino acids and proteins. So in this video, we'll be discussing about the structure, classification and stereochemistry of alpha amino acids. So the 22 alpha amino acids that can be obtained from proteins are subdivided into three different groups on the basis of their structures of their side chains are. Okay, so we told the general structure of an amino acid is C double bond OH, and there is a R side chain and NH2, right? So this is your alpha carbon. So depending upon what is attached, what R is, so the classification has been done into three groups. The first is your neutral amino acids. So some of the examples of neutral amino acids are your glycine, which is G, abbreviated as G or GY. So the structure is this is the structure of glycine. Alanine, for example, abbreviated as A or ALA. This is the structure of alanine. Valine, which is V or VAL. The structure is this is the structure, and leucine is abbreviated L or LEU. The structure happens to be. See, as you are going, the structures are becoming little more complex. That's all. This is the structure of leucine. What about isoleucine? Which is abbreviated I or ILE. Then you have phenylalanine, which means a phenyl group has been substituted in the alanine. Which is abbreviated as F or PHE. The structure is this, and there's a tryptophan. Which is abbreviated W or TRP. So an indole group is present here. So these are your neutral amino acids. Then there are also acidic amino acids, side chain containing acidic group. See, they're neutral for because the basic NH2 group and acidic COOH group are making it neutral. So there's one basic group and one acidic group. Now when you move to a side chain containing an acidic group, there are some more neutral amino acids. I'll show you the chart in the next page, next slide. So these are the important ones. Side chains containing an acidic group. So you have aspartic acid. So aspartic acid is abbreviated as D or ASP. You also have glutamic acid. So which is your abbreviated E or GLE. Okay. So this was about side chains containing acidic group. There are side chains containing a basic group. The classification, third classification. So you have lysine. So you have NH2. It is abbreviated as K, or see, this is basic because there's two basic groups are there and one acidic group is there. That is net result is what you're getting a basic. So there is also arginine. Then you have histidine. So it is abbreviated as H or. So I'll show you the entire chart here. Maybe that will help you. So I will zoom out a bit. So this is your chart of different amino acids. So about 20 have been shown here. So take a look, see the different types of groups that are present. While their basic structure is still the same. See, same, 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 same. What is changing is the side group. What is attached to a side group? See, a same, same. I think proline is different. Other than that, everywhere it is very similar. This is your glycine. Okay. Now, So the amino acids have an important structural feature in common, with an exception of glycine. Whose structure, as we saw, is this one. So this is achiral. Okay. So chirality is also called as handedness. Yes. Okay. It is it means asymmetric. A chiral molecule is an asymmetric molecule. Molecule, meaning that it has no plane of symmetry in it. It has no internal plane of symmetry. And not superimposable. Upon their own mirror image. So except glycine, all the other amino acids are chiral, and all the amino acids have L configuration at the alpha carbon. All the other amino acids, that is except glycine, L configuration at the L configuration at the alpha carbon. So that is, what does L mean? See, this L and D have been taken from their base differentiation lies from glyceraldehyde. So which means they have the same relative configuration as L-glyceraldehyde. So how is that? Let us see. Let R group R be any side chain. So which means your hydrogen is there in the backside. So this, if I have to so if this is your chiral carbon, so I can number them 1, 2, and 3, and since they are going in anti-clockwise, they are your S alpha amino acid. If I do the Fisher projection, that is R group is down and H and NH2 H again. So this is your 1, 2, and 3. So 1, 2, 3 is R, but hydrogen is there in on the horizontal. So therefore R again reverses to S. So your S is the correct configuration. So these are your L alpha amino acids. Okay. Now glyceraldehyde, the structure is why they are same. See, the structure of glyceraldehyde is as follows. Now if you compare the same numberings, if I do, if this is your chiral carbon, it is 1, 2 and 3. So again it is uta with hydrogen atom being at the top, therefore it is S glyceraldehyde or aldehyde or L glyceraldehyde, same as above. See, so this L has been taken from glyceraldehyde, L and D differentiation. So we'll study, when we study your sugars, carbohydrates, more on this. CHO, OH, H and CH2OH. I take this as your, so around this chiral carbon, if I do the numbering, it is 1, 2 and 3, and 1, 2, 3 is clockwise. So but then H is there on the horizontal line, so therefore again I have to reverse it. So this will give you S configuration, same as this. Okay. The next small thing about amino acids is that there are some amino acids which are called as essential amino acids. So amino acids can be synthesized by all living organisms, plants and animals. Many higher animals like ourselves, however, are deficient in their ability to synthesize all the amino acids they need for their proteins. So higher animals require certain amino acids as a part of their diet. Oh, sorry. I made a blunder. Higher animals require certain amino acids as a part of their diet. Okay. So for adult humans, there are 8 essential amino acids. So these amino acids which are to be taken through diet are called essential amino acids. For adult humans, there are 8 essential amino acids. So those 8 are valine, leucine, isoleucine, phenyl, alanine, tryptophan, threonine, methionine, and lysine. So these are your 8 essential amino acids. Okay. So in the next video, we'll go through amino acids as dipolar ions and their isoelectric point. Thank you. See you all.